Welcome and good afternoon. Great to have you all here. My name is Anna Diaz and I'm a lead advisor with Dowling and Yahnke. On behalf of all my colleagues here at Dowling and Yahnke, we're thrilled to have you part of this conversation today. It should be very interesting with Dr. Erica Saphire. So myself, I'm a local San Diegan, born and raised, and frankly, I'm extremely proud of the intellectual capital that lives in this city. We're very fortunate. It's organizations like the La Jolla Institute of Immunology, their incredible research and the critical work they're doing at a very important time in our human history, as you all know. So before I do introduce Dr. Saphire, a little bit about our firm. So we were founded in 1991, which means next year we will be celebrating our 30th year anniversary. When Dale Yahnke and Mark Dowling founded the firm, they were pioneers in the wealth management industry, choosing to be a fiduciary and choosing to be independent, which was quite rare. Today, we manage over $4 billion in assets for over 1,200 clients across San Diego and across the country, providing portfolio management and financial services to those individuals and organizations. We deeply care about the financial lives of our clients as well as their well-being. And frankly, that second part is somewhat what prompted today's conversation. So we hope you enjoy it. We remain humbled by the trust our clients place in us to guide them through these very interesting times. So we really hope you'll enjoy today's conversation. Let's talk about Dr. Saphire. So Dr. Erica Saphire is a professor at the La Jolla Institute of Infectious Disease and Vaccine Research. She was asked by Bill and Melinda Gates to lead a global coronavirus immunotherapy consortium to quickly identify antibodies to combat the coronavirus. In addition, she also led a consortium that united academic, government, and industrial labs across 5 different continents to understand antibody therapeutics against viruses like Ebola, Marburg, Lassa, and others. And just an incredible individual, so thrilled to have her on today's call. A thank you first. So many of you were kind enough to send some great questions over prior to today's webinar. Dr. Saphire has kindly curated her comments based on a lot of those questions. And we want to encourage you to keep them coming during her talk. So you will notice on your screen, there is a Q&A box or comment box. Feel free to send those questions and keep them coming through there. Our team behind the scenes will be aggregating those, and I will lead a Q&A session after Dr. Erica Saphire speaks. Before she begins, I am going to introduce Chris Lee. He is the Chief Advancement Officer with the La Jolla Institute of Immunology. He's going to introduce the organization, and then he'll also then pass it over to Dr. Saphire. So again, we're thrilled to have you here, and with that, we'll pass it over to Chris.
Good morning, everyone, or good afternoon, depending on where you are. It's really an honor to be here and have the chance to address many of you from all over the country, all over the area, and to give you a little bit of background on what the great work that Dr. Saphire is doing, and especially at this very important time. Before we go to Erica, let me take a few minutes to discuss who the La Jolla Institute is as a way to give you proper context. So the Institute is, like the firm that you all are involved with, is about 31, 32 years old. We are a nonprofit organization, one of the best in the world for our work in studying the immune system. We have about 500 employees. Most are scientists. And our budget this last year, as you can see in the graph below, is about $64 to $65 million a year, primarily funded from the competitive grant process through the NIH and other significant granting agencies like the Gates Foundation. And we also have a growing revenue stream for some of our products and ideas that have been brought closer to market. And lastly, the importance of private philanthropy has never been as relevant as it is today. And this has really propelled a lot of the work the Institute's doing in this specific pandemic. So the next slide, please. And as I mentioned, one of the reasons the Institute is probably lesser known than many others that you on the phone might have been involved with or aware of is that we liked our role as being a very focused organization on just studying the immune system. And by just, that's obviously a significant process. It's a significant work. And in this case, we're seeing one of the great values of understanding how the immune system works for when there's a new virus that's affecting so many people. We have the ability through tools and great minds and researchers to understand how that might affect our immune system. Next. Quickly, just to let you know, what our research allows is work on any number of human health conditions. Last year, we started a new strategic plan over the next 10 years, which allowed us to focus our work in 3 key areas based on the prevalence of conditions in these various categories, such as we have a Center for Cancer Immunotherapy, a Center for Autoimmunity and Inflammation, and we look like great visionaries by establishing a Center for Infectious Disease and Vaccine Research, again, the middle of last year. And that wasn't as a result, clearly, of what we knew was coming, but instead just the strength what this organization has, which has really allowed us to hit the ground quickly when this pandemic started to spread. Next. And the last thing before I turn it over to Erica is to put it in perspective. We have about 20 faculty who work in those various areas that I just referenced. And many of them put their attention and their great work and effort tirelessly, in fact, into working on the coronavirus when, again, earlier this year, it looked to be something that was of such great importance. So we have a team just mentioned here below that are looking at various aspects of the pandemic and trying to find solutions for all of us. Obviously, the face you see here in the top row, and you will hear from in a moment, has a significant background in this area and significant work that's already been launched for several months, including great backing from initially the Bill and Melinda Gates Foundation. So without further ado, let's go to the next slide and introduce you to the star of today's show, Dr. Saphire. At the end of the presentation, we're going to put up some information on how you could find out more about the Institute and Erica's work. We're here to answer anything you have or need. And ultimately, if it works, where we're all allowed to eventually see each other in person, we'd love to have any of you out to the Institute who can to see our work in person and to see our Institute operating. But again, thank you for the time today. And we look forward, like all of you, to hearing what Erica has to say. Erica?
Thanks, Chris. Thank you to all of you for joining today. It's such a pleasure to get to speak with you. I wish I got to see you in person. I'll give you an update on some of our research and some of your questions, and the questions that I haven't answered, I will get to at the end. What I'd like to do is give you an update on where we are, what's happening in a little bit in the world, and then more importantly, how our research is going to get us out of our houses and back to what we meant to do this spring and summer and into the next fall. So where we are right now is a continuous burden of coronavirus cases, where we've had a cumulative total of over 2 million in the country and 120,000 deaths so far. The California shelter-in-place order, you can see, happened really early on. And the readout in the number of cases and the number of illnesses, the number of hospitalizations has a lag behind when people get infected. People tend to appear in the hospital 2 to 3 weeks after getting infected. So any behavioral change, societal change, you don't see the effect of for about 2 or 3 weeks. So you can see that when we issued that shelter-in-place order and we all returned home and isolated ourselves from other people, the number of cases still continued to grow, and that's because a lot of these people were already infected by that point. And then with the transmission of greater than one, so each infected person tends to infect at least one, likely more than one other person, cases will continue to grow. There has been some control, but not enough. There are some states where new cases are decreasing. These are For example, in the northeastern states that had a tremendous burden of infection early on and had to introduce some severe measures and keep people home, cases are starting to go down in those places. But there are other states where cases are rising, and in some places that slope is increasing. They're rising faster than they did earlier in the outbreak. And many of these states are places that didn't see the same brunt of infection earlier. And so this is not a second wave. This is their first wave that's continuing. The virus hadn't been in all places in the country at all times. Now, because I'm a scientist, I like to look at numbers on the same scale. If we scale these plots to the total burden of cases, you can see that New York had the greatest burden. So they would top 10,000 new detected cases, for example, in each one of these different days. The dark red line is a 7-day average, and that's divided across a population of 19 million people. If we put up a few other populous states, you can see that cases in these places are now starting to climb as we go out of our houses and interact with people more. This is completely to be expected. If we interact with other people, we don't have vaccines, and we don't have we don't have treatments, and we just don't have enough access to testing, the virus was certainly going to spread. And this is exactly what we see happening, and something that we need to keep an eye on to make sure that we are safe. Now, it has been proposed that the reason we're finding more cases is because we are doing more testing. And while that is absolutely true, at the same time, we're seeing a decline in cases in New York in the absence of new testing, in the presence of more testing. So I think that that is a real decline, whereas I think that what is happening, for example, in our state of California, is a real increase. I think that we need to be careful what is happening in the world. And in other places like Florida, there was an initial decline as people were, I think, scared and stayed home. And as they started to say, well, I didn't personally see a problem, or I must go back to work, we're seeing now an increase in cases and corresponding changes in human behavior. So we look at California just by itself, and we mark the points in time and different cases. We can see here was the number of cases we had when we issued the shelter-in-place order. Cases typically take about 2 weeks to manifest disease from an initial infection. And so this is 2 weeks later from when the California shelter-in-place order was issued. Here are some other time points to mark. Here's Easter, there's 2 weeks later. Here's Memorial Day, there's 2 weeks later. And we're seeing a steady climb now into the summer. Now, what would have happened if we hadn't issued that stay-in-place order and we hadn't had any social distancing? Well, we don't exactly know, right? No one's got a crystal ball. It could have been an absolute uncontrolled increase. It could have been a rapid increase at the beginning. People got smart, decided to stay away from each other by themselves with a leveling off, or maybe something in between. We don't know. There definitely would have been more cases, more deaths, and it's likely that systems would have been beyond capacity. But where are we now? We are in mid-June, and we are seeing a steady rise in cases and a steady impatience with staying at home, and we still don't have the vaccine. So what are we to do about this? Well, we can ask some of the experts. So in a recent New York Times poll of 500 epidemiologists, the newspaper asked them, well, in your personal life, which of the following things would you be willing to now go and do this summer versus several months from now versus a year from now?
Erica?
Yes.
Sorry for stopping you. Do you mind maximizing the slides on the screen and taking some of the extra things around the edges will come off?
Oh.
Easier to watch. Thank you. Sorry.
Is the Q&A showing up? Is that what it is?
There we go. It was the slides were there was a color palette to the side of the slides. It wasn't really on the screen.
That's interesting. That better? I think so. So they asked epidemiologists what they'd be willing to do this summer. And a majority said they'd be willing to go back to the doctor for a non-urgent appointment. Majority of them would bring in their mail. Interestingly, more of them would vacation overnight out of town than get a haircut. And maybe that means that epidemiologists really need a vacation right now, or they've learned to get at-home haircuts. There are a lot of activities that we can see the epidemiologists aren't willing to do just yet, which is send kids to school, camp, or daycare. They would prefer to wait several months or work in a fall, work in a shared office, ride a subway or a bus, or travel by airplane, or eat at a dine-in restaurant. And that's quite interesting considering that we're seeing a lot of restaurants now open up for dine-in business. We're also seeing them with an effort to spread out, closing down streets, for example, to spread tables farther apart from each other. And they think it will be about a year or more before they might be willing to stop routinely wearing a face covering or attend sporting events or concerts or plays. So we are not going to be back to normal for quite some time. And what we're waiting on is something other than herd immunity to give us some defense. Herd immunity is when enough of the population bears immunity through something, either surviving infection or getting a vaccine, that presents a fire break to spread of the virus. And we don't really know how much herd immunity we need for this. It's been estimated that we need about 60% of the population to be immune. And we are not there yet. So on the left is what 60% immune in purple dots versus gray dots looks like. And if we look at cities that have had an abundance of infections, New York City, still only about 20% seem to have antibodies to the novel coronavirus. Stockholm, with fewer restrictions, has only 7%. The rest of our country, just a few percent. So where are we overall? Only a few percent of infections, but it cost 120,000 deaths to get to that point. And so we need something else. And it's not just that the infection is a bit of nothing if you just merely get sick. There are long-term health consequences, and it's taking people months to get over them. So we very badly need vaccines against this virus in order to return to normal life. The earliest estimates for when vaccines might be broadly available is January. Other scientists are predicting it might be summer to fall. Either way, we're going to need to get through the next 6 months to a year, one way or the other. And the other issue is that the important date is not when the first person can go to the first doctor's office and get that first shot. The important date is when humanity is broadly protected. Even once there are vaccines available, they are not going to be in all places at all times. They'll be mobilized to some places faster than other places. And we have seen how quickly the virus can now spread around the world if there's any area that it has reemerged and is recirculating. And even once vaccines are available and are broadly available, there are these other issues. There will be people that haven't been vaccinated. There will be people that just haven't been vaccinated yet. Vaccine's not there yet, or they didn't get an appointment yet. And then there are the groups that can't be vaccinated. So pregnant women, the immunocompromised, all babies under age 1. For some kinds of vaccines, these people cannot be vaccinated, and then for other people, the vaccine simply doesn't take. And so we need to find a way to protect all of these people and find something in between to treat an infection when it has become established. And so an alternative strategy, and this is the one my lab has been focusing on, is antibody therapy. Now, antibodies are molecules shaped like these little Y structures that your immune system makes in response to an infection. If you have been infected over some time, you raise these antibodies. The job of these antibodies is to anchor onto and inactivate the virus itself. They can also recruit immune system help to clear that virus in your body or clear infected cells so they don't turn into virus factories. They are remarkably precise. They have fingerprint-like recognition for a particular place on a particular pathogen. You also raise these antibodies from vaccines, but the ones that exist in the world now, since there aren't vaccines mobilized to most people, are the ones that have come from survivors. And so you can actually look in the blood of survivors what antibodies they have made and see what of them are the most effective against the virus. And here's the issue. There are a quintillion possible antibodies in human sera. It's a tremendous combinatorial array of different kinds of molecules with different kinds of functions. You can give the very best 1, 2, or 3 as a drug. That's called antibody therapy. And you're familiar with some things like that, right? You all heard of antivenom. If you are bitten by a snake, you get an antivenom that is a preparation of antibody whose job is to bind to the toxins in the snake venom and neutralize them to prevent the tissue damage. And with antivenom, you know that it can be the difference between life and death, and antivenom earlier is better than later. It's the same thing with antibody therapy. Antibodies against a virus could neutralize that virus, knock the level of virus down several mathematical logs, and allow your immune system time to recover and clear the rest. There are precision antibody therapies now that are in use against several kinds of cancers that can seek and destroy individual cancer cells that you could not remove with a surgery or find any other way. The lovely thing about antibody therapy is its speed. You can receive that immunity passively moments after IV delivery of those antibodies. And it can last a while. The natural antibodies might last 3 weeks. You can engineer them to last twice as long, but it is transient. It is not a permanent alteration in anything about you or immune system. It is a natural human molecule that you would have made yourself had you had the opportunity. And you can use them either to prevent infection. You can give a frontline medical worker antibody prophylactically to prevent them from becoming infected before there's a vaccine, or you can use them to treat illness, to prevent a disease from progressing to a more severe disease. The challenge is figuring out which antibodies you might like to use for this purpose. So like I said, there's a quintillion possible. And right now there are at least 50, probably 70 different groups that are all racing to discover and deliver antibodies against the SARS-CoV-2, the novel coronavirus. Now, each one of these companies or academic labs is doing something a little different. They might have used a slightly different bait to catch and find those antibodies. That means they'll find different ones. Or they might be using different strategies and different experiments to try to evaluate those antibodies. So they'll be choosing different ones. And they would all like to see their therapies mobilized toward clinic. But there's just a limited number of clinical trial beds and clinical trial dollars. And if you evaluate each of 70 different therapies, you're not going to have the statistical power to understand which of them are good. And you want to make sure that if you're going to put something into a person volunteering for clinical trial, that you're giving them something that is very safe and very effective from the get-go. So which of these competing antibody efforts is producing the right therapeutic molecules that we would want to use in people? And how do we know which molecules are the best? The other issue is that this is a virus that mutates. Now, it doesn't mutate maybe as quickly as viruses like HIV or Ebola, because those viruses don't have any way of proofreading genetic errors. The coronaviruses have some mechanism of proofreading, but it does mutate. And there's new research that shows if you give just one antibody, that virus will happily and rapidly escape around it. You might need a combination of antibodies, meaning deliver at least 2 at once, in what we call a cocktail, to prevent escape. It is harder for the virus to mutate to escape 2 things at once than 1. But if we're putting things together as a combination, we need to find molecules that will work well together. Ideally ones that have some kind of synergistic effect. Well, the binding of the one enhances the activity of the other, or at least an additive effect. And that best combination might come with different molecules from different discovery groups. And we wouldn't know unless we had some way of comparing them side by side. The other issue is that antibody therapy is more expensive than, say, a vaccine or a small molecule pill you would swallow. It's life-saving, but it's more effective. Now, the target product profile for the consortium I'm going to talk to you about is optimistically a $50 dose. Optimistically a $25 dose, we would settle for a $50 dose. But because that is still out of reach for individuals in many parts of the world, we need to find ways of making antibodies that are more affordable. $10,000 a dose won't go very far. We need to find ones that are going to be a few dollars. And to do that, we need to find antibodies that are exceptionally potent. If it is more potent, you don't need to deliver as many milligrams. And so what you can manufacture can be used in more people. We need molecules that are very easy to manufacture and scale up in a production facility so that it's easier to make more of them. That also lowers the cost of goods. We also need antibodies that are going to be very stable, where we can deliver them anywhere in the world and not have them fall out of solution or not have them degrade. We need molecules that are good manufacturing tools as well. And we need to make them broadly available anywhere in the world. It has become very clear that virus somewhere in the world has rapidly become virus everywhere in the world, that a small number of infections in Wuhan spread to the rest of the world. And reintroductions have led to flare-ups, and we see how in gatherings, 10 to 60 people may go home infected. And so we want to make sure that there aren't pockets of the world where virus remains, and that we have something that we can mobilize to treat virus anywhere. To do that, the Gates Foundation asked me to build an effort, this effort, CoVIC, the Coronavirus Immunotherapy Consortium. They asked me to gather all of the therapeutic antibodies being raised around the world and compare them side by side and tell them which are I think are the most effective and the most tractable to delivery anywhere in the world. And so this involves asking each of the different companies or small startups or academic labs discovering these to share their therapeutic candidates so that we can evaluate them side by side. And the point of evaluating them side by side is to do that apples to apples comparison. If each company is evaluating their antibodies and choosing their antibodies different ways, and each company is doing a different kind of experiment to evaluate them, it is very difficult to really understand which molecules are best. You can see that company A will say, I have a really good therapy and I know because I did experiment A. And company B says, I have a therapy I'd like to mobilize and we know it's good because we did experiment B. And company C has a different therapy and they know that's good because they did experiment C. And you're the NIH and you're trying to figure out which one to test in a clinical trial. How do you compare A, B, and C unless they've all been on an equal playing field, run side by side under the same assay conditions? So that is what I have been asked to do during this outbreak, is to figure out which antibodies actually are best, and then to engineer them and figure out how to make them better, how to make them more stable, more potent, better able to be delivered broadly around the world. Now, the idea for doing that kind of large-scale effort came from one that I did against Ebola virus a few years ago. In 2013, which was just a year before the big outbreak, when we always knew there could be one, feared there would be, this is where the state of my research field was. We had therapeutic antibody candidates that looked fantastic in the laboratory, in cell culture and test tubes, but they didn't protect living things. And we had other therapeutic antibody candidates that we knew would protect living things, but they didn't register as successful in the experiments we were doing in test tubes. And so we were at a little bit of an impasse. We don't want to save test tubes from Ebola virus, we want to save living things, but we don't know what experiment we should be trying to do in the laboratory to get there. And the issue, as I saw it, is that we were clearly missing some information. Either we weren't doing the right kinds of experiments, or we hadn't looked at the right set of antibodies. We hadn't looked at enough antibodies to learn the rules. We only found the exceptions. We needed better tools. We needed better samples. The other issue was that everybody in the field was working in a silo by themselves. They were only focused on their one set of few that they had characterized by themselves or evaluating them using just the assays of their lab. Nobody had a broad view of what all of the different antibodies being raised around the world looked like, and nor were the therapeutic candidates being raised in different parts of the world evaluated using all of the different kinds of experiments. And we thought that this was a failing, that we needed to do a better job for global health. That in order to get beyond that research impasse and find some way of making antibody therapeutics available for what I felt would be a growing pandemic, we needed to do three things. We needed to be smarter about how we did this research. We needed to work a lot faster. At this point, that Ebola outbreak had begun. And we needed to find some way of working together that we hadn't before. Now, it makes a lot of sense that you would need to do these three things: smarter, work faster, work together. But in practice, how you put that in place is more challenging. The speed, of course we must work together, but the time it takes to organize into a group could let a pandemic grow. And of course, we think we must work together, but even if the scientists had wanted to contribute their unpublished samples, or the company contribute their new therapy they had discovered for competitors to analyze, their university might not have let them because there's intellectual property involved and a significant investment, and it's publish or perish. It might be difficult for an academic investigator to hand their unpublished molecules and results over to a competitor to look at. So we needed to establish a framework that would enable sharing while protecting their intellectual property and protecting their investment. We needed to find some ways of doing these things at the same time. And so similarly, of course we need to figure out how to do better experiments, but what are they going to be? And if we've organized into a large group, how do we avoid groupthink? We're all familiar with examples where we say, why is that piece of public art so bad? And the answer is always that it was chosen by a committee, right? If we've organized a large group of brains to think about a problem, how do we make sure that we're listening to individual voices and coming up with the right solutions? And if we've organized into a large group, how do we get a very large group to move quickly? How do we do all these things at the same time? And the answer really came down to this, that we needed to do a better job of collaborating, but we needed to do it in a way that still preserved the speed and innovation of what normally drives scientific research, which is competition. The competition between different companies to find a therapy, or the competition between different labs to find a result and publish a paper, is what encourages speed and what drives innovation. That is why we have so many successful discoveries and advances. And so we need to preserve the things that make competition good while allowing a framework for collaboration. And so how are we going to do that? Well, at the same time, we know that it is necessary. In order to solve these major questions, we needed to have a broader set of samples. We needed a broader set of expertise with different people from different disciplines and different kinds of ways of thinking about the problem applied to it. We needed to find a way of comparing samples side by side that belong to competing groups, and we needed to do it a whole lot faster than we were doing it on our own. And so we devised a research study by which all of the samples would be blinded. They'd all be given a code name. So nobody would know whose samples were whose. It would be very fair. It would just be based on their performance and the data. All the samples would come to my lab. We'd give them all code names, and we'd pack identical box sets of the world's therapeutic antibody candidates against Ebola virus and send the identical box out to every lab who did some kind of experiment to evaluate something about the antibodies. So if you were an expert in experiment A, you do experiment A on the set of antibodies. If you're an expert in experiment B, you do experiment B on the same set of antibodies, and same with C to Z. And we'd all met back in San Diego and pooled the large data set together from all these different groups working on the same very large set of antibodies to figure out which antibodies were best, what it was about them that made them the best, and which experiment told us that they were best, so we could revise the research pipeline and do something faster next time. And with the understanding that now the Ebola virus had infected metropolitan areas instead of remote villages and could spread quickly, we couldn't wait for the perfect solution. We needed something now and something better later. And so we devised sort of a tortoise and the hare strategy where some initial therapeutic molecules that looked pretty good could be evaluated and mobilized while we did the larger, more complex study to figure out what was really the better therapy and the right therapy. So we did a fast track and a comprehensive track. And so what we built was a consortium of 44 different groups across 5 continents that bridged academic, industry, and government arenas to do a single comprehensive effort of figuring out what antibodies were best against Ebola virus and how we'd find them better the next time. And so over a couple of years, and this is the data set we generated, where the vertical axis were all the antibodies that people gave us to study from their labs. And the horizontal axis is every single thing we measured about those antibodies. And so because we had a lot of data, we could build network correlation models where we would look at each feature we measured about the antibody and how it linked to other features. If it bound in a certain place, would it inspire a certain function, which things track together. And then we could build a logistic regression model to figure out what it was that allowed an antibody to confer survival. So we can write an equation, which is at the bottom, which is protection equals 1/1 plus E to the whole bunch of terms. And each of those terms has a beta and a chi, and the chi is some feature of that antibody, and beta is the importance of that feature. And we're visualizing that for you in the bars. Where blue is a good thing, if antibody does that, it's more likely to protect you. And red's a bad thing. If antibody has that feature, it's not any good. A long bar is a very important predictor of success, and a small bar is a not so important predictor of success. And if we do these 17 things, and we can do high throughput in plastic and test tubes really fast, we can predict whether or not that antibody is going to be protective with a 96% accuracy. And that's pretty good. But what we found was that we had indeed been doing some of the wrong things. It used to be that what the field was focused on are these things that say previous focus. The one in the middle was whether or not it inactivated a certain kind of model virus. And we also looked at where the antibody bound and how tightly it bounds. We thought those would forecast success. But they're not all that important. Instead, we found better ways of measuring the ability of an antibody to physically inactivate a virus. And we found a lot of new features that hadn't been explored before. And those are all the ability of a part of the antibody called the Fc, that's the base, to inspire the immune system to come and clear the virus from the infected cell and clear the virus from the body and clear infected cells as well, so they stop being viral factories. That the ability of that antibody to not act alone, but the ability of that antibody to inspire the power and complexity of your human immune system to clear itself of that infection was just as important as what we had previously thought to look at, which is the mechanical activity of that antibody. And so after a couple of years, this group wrote 78 peer-reviewed papers. We granted 10 PhDs based on the research we inspired. We revised the research pipeline, so now people do different things and analyze antibodies in different ways. And we produced the information we needed to put 6 new therapies in place where there weren't any before. And you may have read about this in the paper, where suddenly there was a treatment for Ebola virus when there hadn't been before. And that is the best one, is this cocktail here. Now, this is what I do. This is the specialty of my academic lab, which is the precise molecular imaging of things that are 100 angstroms across, so 10 to the minus 10. They're impossibly tiny. They used to be submicroscopic. By making the map of the submolecular structure and actually peering in, we can see the ribbon of the polypeptide chain, the genetic sequence the virus builds. We can see how the antibodies anchor onto those proteins and how they work. And so that kind of information from those very powerful microscopes in my lab and the x-rays that we use help us directly see how those therapies work. And seeing how they work tells us how to choose them. Now, that was the program that we ran, and we were busy evaluating some of those novel therapies that came from that when this virus emerged. And that was just about 6 months ago when this novel virus came onto the scene and upended all of our lives. And so the Gates Foundation called and asked if I would run that same consortium for the antibody therapies they needed to mobilize very rapidly for coronavirus. And that gave birth to this consortium here, where again, we are gathering the therapeutic candidates being raced forward to the clinic by multinational corporations, small startups, and academic labs, comparing these competing groups' antibodies side by side in a blinded fashion to figure out what is best, what we think should go into people, and how we can make it available broadly around the world. When the antibodies come in, just like for Ebola virus, they'll be blinded, they'll all be anonymized, and that is to encourage participation and to make the data very fair. We are distributing them to key partners around the world that are going to be running different kinds of experiments to analyze how they recruit immune protection, where they bind, how well they bind. And importantly, we're going to be putting all of this information into a publicly available database, which is going to be available on the web real time, so that researchers around the world can see what we are learning about all these antibodies as we build that. And that's very important, because if we've gone to the trouble of finding ways of convincing these 50 to 70 competing groups to partner together, and we've gathered all of the world's therapeutic candidates in one place, there's an opportunity to learn something that's going to be a lasting body of information. And there's a benefit to these companies too for participating. They can use all of the data that we generate, and many of these are techniques to which they don't have access. They can use all of that data for their own investigational new drug filing. and as an independent verification that we think this is a good therapy and should go forward. Our goals are these. The first one is we need to put products into people. We need to find combinations of at least 2 human antibodies that we could use as a therapy against SARS-CoV-2. And we want to optimize them to minimize the likelihood the virus will escape them and to make sure that we can successfully deliver them. But with my academic hat on, I want to make sure that we are taking advantage of this opportunity. If we have the hundreds of therapeutic candidates side by side and we're looking at them, we could learn a lot of information that's going to help us next time, because this virus is going to be here a while. We could probably expect seasonal return of this virus or another coronavirus. We need to understand which of the antibody therapies in this arsenal we have built are going to be responsive against newly emerging forms of the virus as it recirculates every season. We'll be able to, by looking at the world's leading candidates side by side, figure out how antibodies can defend against this virus, what makes one more successful than the other. When we make combinations of 2, what adds to potency? Where can we get synergy? What determines how these antibodies target the different parts of that spike on the coronavirus? And the antibodies that we're looking at, how closely do they resemble the ones that we're hoping that vaccines will inspire? How do therapeutic antibodies differ from the broad range of antibodies that will be elicited in people when the vaccines start to become available? Our use cases are these. Saving lives is our highest priority, and our initial focus is going to be on treatment, to find people who have been infected, who have mild to moderate disease, and prevent them from progressing to severe disease. So we'll look for patients that may have a likelihood of progressing to a more severe disease, and they would be prime recipients of protective antibody therapies that could help them along. We'd also like to use some of these antibodies prophylactically, especially as there's no vaccine available yet. And this could be to protect frontline workers and responders, to protect high-risk groups, including family members of people who have become infected, pregnant women, and to ring reemergence and outbreaks, to treat places where the virus has reemerged to prevent it from establishing a new pandemic. And we're very much in the thick of it, where we have done the hard work of the negotiation and the consortium building, and the samples are starting to arrive, where we are now blinding them and doing that structural biology to mobilize, to understand how they work and mobilize these things more broadly. So that is terrific. We're going to get therapeutic products. We're going to get a lot of information. There are things rushing toward clinic now. We're going to be evaluating those and seeing if there's a second generation therapeutic that's going to be better later. But one issue that we are worried about and want to keep an eye on is how we make sure that these antibodies remain responsive if the virus changes. Right now, there's a tremendous quantity of human-to-human transmission, with millions of cases around the world, and each of these infected people interacting with others. And every time that virus grows in a person to millions of copies, or they transmit from one to another and in different places, that's a lot of opportunity for the virus to acquire mutations and acquire escape. So we want to keep an eye on that, especially as the cases, bottom right, are growing at a steady level, and the virus is now essentially anywhere where there are people. One thing that we particularly want to keep an eye on are whether any changes emerging in this virus are escaping the antibody therapies that we have. And a recent finding is this, and this is work from Betty Korber's lab that was now posted in an online repository called bioRxiv, that a single point mutation at position 614 emerged in March and became fixed and established throughout the world. So the orange graph are cases of people infected with the original virus that emerged from China, that we call that D614. It's got a D amino acid at position 614. But that single point mutation arose, and that's the blue virus, the G614 virus, and it's rapidly become dominant with the span of a month. So for example, cases in Europe and in North America rapidly started seeing a lot more cases of G than ever before, relative to D. It became dominant. And in England, they were nearly completely infected with the G-type virus. And the G-type virus caused a second wave in Taiwan, where you can see that they emerged and cleared the D-type virus, but then the blue one, was introduced there, which caused another wave of cases. So what is this change, and what does it do? Well, we absolutely need to find out and keep an eye on. And the eye that we are keeping on this is this tool here. This is one of the most powerful microscopes commercially available that we bought for my work at La Jolla Institute for Immunology. We have been building it over the last few months, and we're going to fire it up tomorrow. This is called the Titan Krios. It is 11 feet tall. So it is not the little microscope that you dissected a frog in high school biology class. This is 2 times as tall as a person. And it shoots an electron beam that is 300,000 electron volts down that central column in the working sphere. You can see in the back, they're building all of the computers that run it, the servers. And the goal of this is to peer at unprecedented resolution to visualize the very precise single mutations and single amino acid changes and loss or gain of single hydrogen bonds. Because even those very small changes can affect how the virus transmits across people, affect whether this drugs will work or not, or whether the virus will propagate. And so this is an image from a microscope like that of the spike on the coronavirus. This is the molecule that the coronavirus uses to attach to your cells and drive itself in. If we peer down into the structure of that molecule and we look at its atomic assembly, we can see that mutation that arose and has now become dominant. And that's that red amino acid there, where it was an aspartic acid and it switched to a glycine. And that change of loss of that aspartic acid and switch to that glycine, we think, has destabilized that portion of the virus and allowed it to do some different things. Okay, so what does that mean? What does it mean for how the virus grows? Well, this is new data from my lab. We made versions of viruses, of recombinant VSV, bearing that spike. And we looked at how well they would grow in different kinds of cells, comparing the old spike to the new mutant spike. So we looked at them in a monkey cell line and different human cell lines that did and didn't have a key enzyme that that virus needs to get in. And what we're showing you is every time there's an orange circle, that's the growth of a virus, a different replicate of an experiment, the virus bearing that old spike. And then the black T-shape in the center, the middle line is the mean, and then the upper lower one's standard deviation from the mean. So you can see the clusters of how well the virus would typically grow in these kinds of cells bearing the old spike. When we make that single amino acid change, introducing the new spike, just that one small substitution, the virus grows better. In fact, it grows 2 to 3 times to a higher titer from that one single change. So that's what happens in cell culture in the laboratory. What happens in people? Well, this is other data from Betty Korber's larger collaboration in bioRxiv, that we can measure how much virus there is in people by the cycle threshold of diagnostic PCR. So what this is it measures the number of cycles of amplification you have to run in order to detect a virus in a person. If you don't have to run so many cycles, there's more virus. So you have to run a lot of cycles of amplification, there's less virus. So the new virus is blue and the old virus is orange. And you can see there's a tremendous range. Some patients don't have very much virus, some are growing very high viral load. But if you look at the mean of blue is lower than the mean of orange. There's fewer cycles of amplification needed to detect the new virus than the old virus, G over D. So not only does that virus grow on higher titers in laboratory and cell culture just by changing that one amino acid, that one molecule, it grows to a higher level in humans too. So are we in trouble? I just told you the virus grows 2 to 3 times as fast. I don't think so. Not yet, and here's why I don't think so. So if we look at the convalescent sera, this is from the blood of a person in San Diego that survived coronavirus. He was infected in early to mid-March. we can see how well the antibodies in his blood can neutralize the new versus the old virus. So I'm showing you here a neutralization curve. So orange is the old virus, blue is the new virus. On the vertical axis, we're measuring for you the relative ability of the virus to infect cells. So at the bottom is no relative infection, the top is complete infection of the cells. And on the x-axis, we're looking at what dilution of the antibody gets us there. So on the left, 10 to the 0, that's antibody straight up. Next tick over, a 10-fold dilution, 100-fold dilution, 1,000-fold dilution. We're going to log scale dilutions in the antibody to see how little antibody can still inactivate that virus. So this is log dilution of the serum. At that end is antibody straight up. This end is antibody highly dilute. And you can see the curve of less and less antibody allows more and more virus. That's how we look at the ability of an antibody to inactivate a virus. So you can see when the antibody is present in decent level, that's the left and middle part, the relative infection is 0. The antibodies are finding, taking care of, and inactivating the virus. But what we want to compare is what happens with the old versus the new. And so we typically look at two places where half the virus is neutralized and where 90% of the virus is neutralized. I've got that in the wrong place. But we want to see the points where comparing the old and the new. And you can see the new is to the right. It is just a bit easier to neutralize the new virus than the old virus. And that's just one person's blood. If we look at six San Diegans, they're all infected about the same point in time when both viruses circulated in San Diego. For every single one of them, the blue graph is in the same place or slightly to the right of the orange graph. You need the same or less antibody to neutralize the new version versus the old version. And that's really interesting because we see really clearly in cell culture and in real human infection, the new virus is fitter. It grows 2 to 3 times better. So even though it's fitter and it's growing better, your immune system can still take care of it in an equivalent way. Now, we don't actually know which virus each of these 6 San Diegans was infected with because both D and G circulated in our city at that same time. So they could have been infected with one, they could have been infected with the other. But what we see overall is that even though that virus is fitter and grows more robustly, you don't need more antibody to take care of it. So that's good news. Viruses carrying that newest spike that emerged and became dominant in human-to-human transmission around the world are not harder to neutralize. Now, the limitations are that's sera of 6 people. We need more studies to find out if this is true for the sera elicited by vaccines, because all of the vaccine development started when only that orange D virus was in the world. Now, I think they probably will, because remember, there's a whole array of antibodies in your blood. There are millions. And if 1 or 2 of them are knocked out by that change, you still have the rest. So I think we're still in the clear. And the other thing is that even though the virus has changed to become fitter and stronger, it's not more deadly. There's no worse patient outcome. So if we look at hospitalized people in Sheffield, England, and we can see that over a point in time from early in March to early in April, left to right, a lot more new virus, the blue virus, became established compared to the old virus, the red virus. But they could sequence the virus of each patient coming into the hospital, and they could sort the patients into those infected with orange and those infected with blue. And then they looked at the severity of the infection, about whether that patient could be treated as an outpatient, meaning a more mild disease, whether they needed to be hospitalized as an inpatient, or whether it was very severe and had to be admitted to the ICU. And overall, there's no significant difference, except that well, overall, there's no significant difference we can see on this human level. So the virus is clearly fitter. It has become more effective at infecting cells and making more copies of itself. But it's not more lethal, and the antibodies that we make can still take care of it. So did we dodge a bullet? Well, we may have, but we shouldn't really celebrate yet because we don't know what will happen next. Now that the virus has become a bit fitter, will it acquire another mutation that, for example, enhances affinity for receptor or something else that allows it to evade some aspect of the immune system? It could acquire another mutation on top of this one. What makes us concerned is that a mutation at this time point became globally dominant a month later. So as the virus acquires substitutions and mutations, we do see they become established. There's a tremendous amount of human-to-human transmission happening in the world. It still is there, and there will be for a long time. We're not going to have a vaccine for 6 months to a year. Even when that vaccine is available, not all people will have had it at all times. We're mobilizing antibody therapy. And this is what we need in order to get out of our houses and back to education, employment, and human endeavor. We need all of these things. We need antibodies to treat people that can't receive a vaccine or haven't received a vaccine or in whom a vaccine won't work. We need vaccines for broad administration. We need treatments for those who have become sick anyway. And we need to find better and faster ways of discovering what we need, de-risking these potential therapeutics and evaluating what them are. We are here. We can expect there to be more cases and more infections while we get these immune defenses mobilized. And so we need to be vigilant out in the world while we try to develop these necessary immune defenses and deliver them to clinic. They need to be well evaluated. They need to be safe. They need to be de-risked. But in order to get out of our houses, we are relying on a couple of aspects of immune defense. In order for us to continue really as a human race with our human endeavors in education and employment and social interaction, we are relying on the ability of our immune systems to defeat a virus, the ability of our immune systems to respond adequately to a vaccine, the ability of antibody therapies to support and provide that kind of immune protection. That without that human immune system, we would not survive. You fight off pathogens every single day and you have no idea because it just happens without your knowledge. Your immune system can find and destroy individual cancer cells before they've established and found a tumor. And the different areas of the study of the power and complexity of that human immune system have traditionally focused in different ways, infectious disease or in cancer or in basic understanding of inflammation and how that might get upcycled or overactive to cause autoimmunity. But these different areas of immunology influence each other. That some of the antibody treatments mobilized to control inflammation can now be used to control severe COVID disease. This is a clinical trial going on. Some of the things we learn about infectious disease triggers show us how viral or bacterial infection causes cancer or causes autoimmunity later. And so the benefit of working at a focused think tank on human immune system and human immunology, as I do, is the ability to learn from investigators and labs working in these different areas of human immunology and what we can learn from the basic information gained in the study of autoimmunity that could help us understand something about how we might harness the ability of the immune system to work against an infectious disease, and things we learn about infectious disease that influence how we might develop different treatments for other things, cancers or inflammation. So this is where we work. It's a lovely place, and I'm happy to answer any questions that you may have about the work that we're doing or other things about the virus in the world.
Thank you, Dr. Saphire. Fascinating. Gosh, fascinating information. And first of all, we recognize it's right about 12 o'clock. Dr. Saphire, we still have a couple more minutes. We'd love to do some questions and answers with you. Thank you for a lot of you who have been sending questions along the way. And so we'll do probably about 15 minutes or so of additional questions, and we'll kind of take it from there. So they've come through in a few different forms. And so first, if you don't mind, Dr. Saphire, we'll start more around safety. Mm-hmm. So a couple people have said, we recognize and are excited about some of the sciences that are progressing both in antibodies and, eventually in vaccine. Until then, there's risks. And so, and different demographics and different questions. And so the first one is around, given the reopening, if you're over 65, what activities from your perception are considered high risk and low risk? And a very similar question was asked around students and colleges who, as they revisit, right, starting in the fall as they get back on campus.
Yeah.
Any comments around that piece?
The main thing which is risky is interacting with other people within a close distance. The most common route of transmission is respiratory route, droplets, people within a couple of feet of each other, and especially indoor crowded areas with poor air circulation. And so activities that would be low risk would be things where you are distant from other people. High risk would be things where you are crowded in with other people. So I would say that airplanes and theaters and crowded indoor spaces are higher risk. Outside in the sunshine in a place that's a few feet or more feet away from other people would be lower risk.
That's helpful. That's helpful. So on the antibody side, exciting to hear some of the progress we made on the antibody work. Is there any demographic or particular group of people where antibodies are not going to be an option?
The good thing about antibody therapy is that it is essentially de-risked. We can engineer ones that avoid potential disease enhancement that you might see with other kinds of treatments and therapies. You can deliver antibody to somebody that is immunocompromised or unable to make a strong immune response to themselves. And so a neutralizing antibody therapy tends to be something that is more broadly applicable and broadly effective than other things. It is also typically a safe molecule because it is very precisely targeted toward the virus. And we have ways of ensuring that there's no cross-reactivity to something else. So it's in general a pretty safe thing. The obstacle has traditionally just been cost and availability. The ability to manufacture enough antibody and the ability to make it affordable. And so that's where the Gates Foundation is stepping in to ensure that people can receive this kind of therapy regardless of their ability to afford it.
Yeah, that makes sense. So the education that you gave us on the D and G strain was very interesting. So a couple questions came in along those lines. And so can a person who is infected with the D strain can then recover and then still catch the G strain?
Probably not. So this is one tiny minute change. It changes the virus's behavior, but your immune response is what we call polyclonal. You will make many antibodies. You will make also T cells. Your immune response has multiple arms to defeat a virus. This one small change may knock out 1 in a million antibodies, but you still have the other 999,999 to work. So I don't think so. I think your immune response will protect you from a subsequent infection in the near future. Now, the worry is actually the longer term future, that for coronavirus infections, immunity seems to wane. For some viruses, like your one polio vaccine, you will have immunity the rest of your life. But we see these coronaviruses come back every couple of years. And in a lot of people, the immune response mounted to a coronavirus infection tends to go away after 1, 2, or 3 years. So you could recover from D now and be reinfected from G in 3 years. But that's not the D versus G difference. That's a 1 year versus 4 years from now difference.
Okay. So one of the other questions now is more on the spreading of the virus. And so how that's happening. A few were curious around children.
Mm-hmm.
So do we have a sense of how often children are the non-symptomatic spreaders of this virus? And how is that probably impacting decisions that individuals are making and schools, et cetera?
Yeah. Well, there was initially interest in that question because it looked like children didn't get sick. And they didn't get sick, how would you know if they were infected or not? And so would they be, and you know that kids, maybe are just good at washing their hands and not picking their nose as adults. But it looks like from data gathered in Europe where they reopened schools and sent kids back that it's not as feared, that children do not as frequently transmit virus to adults as adults to adults. It doesn't mean it'll never happen, but it's less common than from an infected adult. And that may have to do with different parts of the respiratory tract being infected to a greater or smaller degree. That's something that we do definitely want to keep an eye on, because we know in schools we have a couple hundred kids in the same building, and then they go home and mix with different families every night. And some of those families are multi-generational. So we want to make sure that we do what we can to keep the kids apart, get fresh air circulating in the schools, have adequate testing, or find ways of expanding the classrooms and teaching them outside, or getting the kids a little further away from each other, putting them in masks. I'm not quite sure what all is going to be done yet, but we'll need to put a lot of thought into that.
It's not an easy one, for sure.
No, of course not. And yeah.
So can we talk about vaccines a little bit? So if you could just comment, because you covered that a little bit too early on in your talk, but if you could comment on just the relevant difficulties with vaccine discovery and approvals, you know, getting global reach. Could you speak to some of that process and how you think that will evolve?
Yeah. So there's a lot of testing that goes into vaccines, and most things tried will not make it all the way to the finish line. There are 100 different efforts right now, and that's very hopeful, because it means that many shots on goal, one or more, are going to succeed. Some of the risks that we have seen in earlier vaccines for respiratory diseases are occasional evidence of enhanced disease instead of mitigated disease. Now, a famous example is in an attempted vaccine, maybe 50 years ago, for respiratory syncytial virus. Respiratory syncytial virus is a severe seasonal winter infection that can be lethal in premature newborns, and almost all of our kids get it. You probably remember your kids having been really sick when they were young and maybe turning blue. An initial vaccine wasn't successful because it was made from virus that they tried to inactivate. And the inactivation procedure, and I think it was formalin treatment, popped all the little spikes in the surface of the virus into a different shape. And that different shape allowed them to infect cells more. And so that vaccine, which was ultimately trashed, in a couple of kids caused worse disease instead of better disease. And so that's some of the examples of ways in which an experimental vaccine can not work out and be shelved. We now know what we didn't know 50 years ago. It was because we have structural biology like I do and these kinds of microscopes, we could actually see what happened to the surface of those spikes in the surface of the virus. They had no idea 50 years ago. They just knew that for some reason this vaccine made disease worse. Now that we have these better tools, we can actually physically look at the vaccines being made and make sure that the molecules are the right shape. And that will mitigate some of the risks that we might have from different kinds of things being tried and developed. The other risks are maybe they just might not work, right? They might not inspire a strong enough immune response. We're going to need a lot of testing to make sure that we get the kind of immune response we want, and that it's extremely safe.
Thank you. We'll do one more question if you don't mind, Dr. Saphire. And for all of our guests, a lot of additional good questions have been coming in. The La Jolla Institute of Immunology has kindly offered to answer those questions directly. So we'll make sure we get back to all of you on those, on the outstanding questions. We did cover a good amount though today. Let's talk a little bit about mortality if you could. So you shared it with us some stats in the beginning, around 117,000 deaths on 2.1 million cases in the country. Would kind of equate to about a 5.5% mortality rate. So when you compare that, you know, what our country is experiencing relative to maybe other less developed countries, why is the mortality rate similar when you would argue we'd have a more advanced healthcare system here?
The mortality rate measured is it's a fraction. It's numerator over denominator. It's the number of deaths of the number of cases. And if you have more testing and you detect more cases, your denominator is going to be bigger and your percent mortality will be smaller. And the issue is that early in an outbreak, if there isn't enough testing, you only find these severe cases because you see them, they're very sick. You don't see the asymptomatic or the mild infections of people that seem to have had some kind of cold. And so if you don't count them, you don't get an accurate estimate of the actual mortality. And that's why those numbers vary so much by place and vary so much. They look worse earlier in the outbreak than they do later in the outbreak. And so I think the difficult message for public health has been that changing number and our apparently poor understanding of exactly what happened. And the other issue is this, that, you know, even though 99.9% of young people will be okay, someone is going to be that 0.1%. And it's not just whether you will survive or whether you will perish. People get really, really sick. And there are long-term complications where it's taking weeks and months for some people to get better. So it's not to be taken lightly.
Thank you. Well, Dr. Saphire, thank you so much for the time that you spent, and frankly, all that you and your staff are doing. Incredible work, and again, in a very important time. And thanks for the additional time you gave us on questions and answers. To our guests, thank you so much for joining us. We hope you found this interesting. We are also excited to share that we are going to have an upcoming webinar in July, on July 9th. We're going to have Marco Biamonte, who is the founder of Drugs and Diagnostics for Tropical Diseases, will be speaking with our group and really discussing testing. So taking this topic, going a little bit deeper on the testing area. So reviewing kind of different antibody and antigen tests and their potential applications. So we hope that you will join us for that. You'll receive an invitation from our team coming up shortly. Thank you again for your time. We hope you enjoyed the presentation. This ends our webinar, so hope you all stay healthy and be well. Thanks.